um, turn on your camera. I'm waiting while I am going to be sharing my screen, and let me know if you can see my screen. Hello.
Yes.
Yes. Okay. So guys, this is interactive class. So I definitely need you to speak, to answer my questions, to be present within this class. Okay. So you are paying me money so you can be taught. So this group is mixed. We have medicine and dentistry in this group. So let me quickly explain what's going to be happening throughout this semester. So first for medicine. For medicine, we have 3 SATs this semester. The first self-control test is going to be on week 7. Typically, it's on week 7, but sometimes they put it a little bit earlier, maybe week 6, because there is a holiday on week 7. So this is why they keep moving it, 6th or 7th week. The second SAT is going to be on week 9. And the third self-control test is going to be on week 14. Judging from the numbers, you can see that the biggest SAT for medicine is on week 7, right? So we have 7 weeks of preparation for this particular self-control test. In my opinion, this is definitely the most difficult self-control test out of the 3 of them. So what happens if, as a medical student, you get 60% on all 3 of them, right? So each self-control I passed. What happens? Your final examination will look like this. Instead of picking 3 slides, you will only pick 1 slide. That's it. Okay, so you will pick that slide and you will explain it. So I personally think it is a very good advantage. I think it's worth it to study for those 3 self-controls throughout your semester. That way your anatomy will be very, very easy. Okay, so this is why please study with me so we can succeed in this part. Now for dentistry, we have a couple of students of dentistry here. Closer to next class, I will switch you. I will put you in different groups because I want to separate you. Just this class is important that you are together. For dentistry, you have 2 self-controls. The first self-control is on week 7 as well. Again, they change it, maybe week 6, maybe week 7, we do not know. And the second self-control is on week 14. So basically for dentistry, 2 self-controls of medicine are put together into 1 on week 14. What happens for dentistry? If you get more than 80%, as it was last semester, we check again this semester if the rules have changed or not, but if you get more than 75 or 80%, I'm sorry, I don't remember for dentistry exactly, on both of them, you're exempted from histo completely. You have no histo in your final oral. So this is why for dentistry, it's really good to study histology. So that way you can definitely just get rid of it, forget it, and not have it in the oral whatsoever. Okay, for medicine, unfortunately, your future doctors, unfortunately, you will either way have 1 or 2 or 3 slides, depending on how many SATs you pass. Lots of students take this approach where they say, well, I mean, I have exam either way, so why even bother studying for the self-control tests? This is not a good approach. Why? Because you're forgetting that you have anatomy to study for, physiology, and biochemistry, and also embryology. You do not want to have all of the things in the final exam, okay? So this is why, please, every class before the class begins, we will have a mini test where I test you either self-control style or oral question styles, but ask anybody, every single question I ask you, they somehow appear in the exam. I do not know how does this happen. Maybe because I learned to think like the anatomy department. So that's why whatever I like twist my mind to ask you, somehow Professor Anto always kind of syncs with me on that level. So they always ask you those things. So as you know, what I wrote for you in the groups, those students who succeed in my course, so for self-controls, for all questions, if I see that you participate, if I see that you answer my questions, you're prepared for every class, our self-controls that you're having with me, the score is more than 90%, all of the embryo course is for free. The entire embryo course is going to be for free. I personally think it is a very good motivation for you to study. You're saving yourself 30,000 forints. So I think it's a lot of money. So this is why you should and you must and you ought to study this part. Okay. So that being said, let's begin. Okay, so here we have the histology first SAT part 1. Now, those students who are dentist students, this is a bit tricky part for you because the first part that we're talking about will not be in the self-control test, but it will be in the final oral. So everything what we're learning right now is in the final oral, assuming that you did not pass the ACT, that you got less than 75%, okay? So this is why you need to know this just to be sure that it will not appear in your oral exam, okay? This is very weird system they have, that you are not asked this as a dentist in the self-control, yet you are asked this in the exam. Medicine, this is your self-control test. It begins from now, okay? So this is the part that begins from now. So let's begin. Oral cavity is the first thing, so this is pretty much straightforward. Oral cavity refers to the internal part of the mouth. We have two anatomical spaces here, the oral vestibule and the oral cavity proper. Where do we have the oral vestibule? Oral vestibule is a space which is located behind teeth, but anterior to your, behind lips, but anterior to your teeth. Okay, so when you brush your teeth, where do you put the toothbrush? You put it into the oral vestibule. Okay, so I repeat, oral vestibule is a space which is basically between the inner part of the lips and the teeth. Or in other words, posterior to your lips and anterior to your teeth. What about the oral cavity proper? Oral cavity proper is a cavity which is everything behind your teeth. Everything posterior to your teeth is the oral cavity proper. Which kind of structure is present within the oral cavity proper? This is going to be the tongue. Okay, so tongue resides within the oral cavity proper. Now, this is just blah, blah, blah. I'm pretty sure everybody knows this. So which particular structures are inside the oral cavity? We have lips, cheeks, tongues, teeth, gingiva. What is gingiva? Who knows?
The gum.
Gum. Okay, so gingiva, this is the gum. What is gum? Okay, so this is this red area right here, right? Pinkish or reddish, this particular component right here, which kind of hides the bone, where the roots of the teeth are present. So in everyday English, we say gum, but as a medical student, you're gonna say gingiva. So make sure that you are familiar with this word, okay? So gingiva. Palate, anybody knows? We actually have two types of palate. We have the hard palate and the soft palate. Who knows where's the hard palate? Or soft palate, any kind of palate.
The roof of your mouth.
Very good. So which palate is this? Hard or soft? Probably, hard. If you touch it with your tongue, it is
hard.
Hard. Okay, so hard palate, this is the roof of your oral cavity. It is the roof of your oral cavity proper. So if you kind of scroll it with your tongue, right? So you feel that it is very, very hard. What about the soft palate? Like if I open my mouth right now, you will see an arch there, right? And uvula hanging down from that arch. And behind this, there is another arch like this as well. So all of those structures, this is a soft palate. So with my tongue, I cannot reach it because it doesn't roll that way back, but this is a soft palate. So everybody is probably familiar with the structure, right? So the two arches, if I open my mouth, and the uvula hanging down. Salivary glands and tonsils. Anybody knows which kind of structures are tonsils? Like, what will be their function? Anybody knows tonsils?
Kind of immune system?
Perfect. That's the perfect answers. Tonsils belong to immune system. And immune system has another name. It's also known as the lymphatic system. And we will learn about it, this self-control. Immune and lymphatic system, this is the biggest component of this self-control test. Majority of questions will come from there because it is very, very important. So tonsils, those are the structures, little tiny organs, little tiny collections of tissues, and they belong to the immune system. Inside of the tonsils, you will find lots and lots and lots of lymphocytes. Okay, so they're filled with lymphocytes. In your oral cavity specifically, you have 4 pairs of tonsils. We will learn about them this SAT as well. Okay, so 4 pairs of tonsils. How many in total is that? 8. They form a ring, and we call this ring Waldeyer ring. It's very important in anatomy and in the histology. Okay, so this is what we call Waldeyer's ring. Waldeyer's ring is the first protective, guard. This is the first area of protection of your body. Think about it. What do you do 24 hours a day? You constantly eat, drink, chew on your pen. You constantly introduce something to your mouth, right? And what do you constantly introduce something to your mouth with? It's the bacteria or viruses or pathogens or fungi, parasites, whatever. So this area right here is the entrance to your entire organism, to your entire body. So of course, we need something which can protect us from anything that comes there. And what is that something? Tonsils. So I'm not sure if you know those people, but I have lots of those people in my circle, which are always sick. I mean, they were just sick for 2 weeks, 1 week passes, they're already sick again. Have you ever met those people? So if you actually ask them to open their mouth and be like, can I look at your tonsils? Their tonsils are enlarged beyond like level, okay? So they're visible with the naked eye. What does that tell you? That tells you that their tonsils are filled with the lymphocytes. So their tonsils are constantly active because there is always some kind of infection going on. Okay, so this is why, again, we'll talk more details about it. Just want to get your attention that tonsils belong to the immune system. Now, finally, for the histological component. What is mucosa? We talked about it last semester. Those students of mine, I taught you. Mucosa, what is it? Who remembers? It's like a layer that covers or lines some organs, but what does mucosa consist of?
Loose connective tissue. Oh, connective
Loose connective tissue will be under it. So under the mucosa, we have loose connective tissue, I agree. But what is mucosa itself? I'll help you. It's 2 layers. 2 layers make up mucosa.
Lamina propria
and epithelium.
basal lamina.
Perfect. Lamina propria and epithelium. So the first layer of mucosa is epithelium. We talked about it in the first histology course, right? The first self-control. Epithelium is the most important thing. This is the thing that covers or lines everything. What is the structure on which epithelial cells are sitting on?
Lamina propria.
Before that?
Basement membrane.
Basement membrane. Epithelial cells are sitting on basement membrane. So right here, probably between. And under the basement membrane, we have the structure which is called lamina propria. I'm going to put it as LP, but it is called lamina propria. What is lamina propria? What is it? What is lamina propria? It is connective tissue. It is connective tissue. But what is so special about this connective tissue? What does this connective tissue has that normal connective tissue does not have?
Lymphocyte.
Lymphocytes. Okay, so lymphocytes are present within lamina propria. This question is a failing question of Professor Antal. What is mucosa? That's his question. Again, what is mucosa? Mucosa is a layer, okay? This layer covers organs or lines organs or protects them, whatever word you can come up with. Histologically, mucosa consists of two layers. The most superficial layer is epithelium. Duh, everything superficial is epithelium. But what is under the epithelium? Connective tissue. But we are in the GI. This is the gastrointestinal tract, the first thing of which is the oral cavity. What did we just say? This is the entrance point. We need protection from all of the future potential pathogens. So this connective tissue has to be special. It has to have lymphocytes. How do we call this particular connective tissue with lymphocytes? We call it lamina propria. So a statement for you. Tell me if it is correct or is it incorrect. Throughout entire GI, lamina propria can be found. Throughout entire GI, lamina propria can be found. Correct or incorrect? Can we find lamina propria everywhere in GI, or will there be places where there will not be lamina propria? Guys, participation is really important.
Everywhere.
It will be everywhere.
I think it's everywhere.
It will be everywhere. Very good. Lamina propria will be found everywhere in the GI. I will tell you why. Okay, yes. Oral cavity, definitely lamina propria because entrance point. What is after oral cavity? Esophagus, right? Still, whatever I have ingested is in my esophagus. Do I need protection? Of course. Lamina propria has to be there. Stomach. Still it is in the stomach. The same thing is in the stomach. Do I need protection? Yes, lamina propria. What is your GI? It's one tube. Just very, very coiled, very, very weird looking, but it is one tube. What is the entrance? Oral cavity. What is the exit? Anus, right? Still, it is a tube. So whatever you have ingested has to pass through this tube and then come out. Do I need protection from that something? Of course. So this is why lamina propria is a connective tissue which will be present throughout entire GI. It doesn't matter where, stomach, esophagus, small intestine, large intestine, rectum, anus, oral cavity, it does not matter. Whatever is under epithelium will always be lamina propria. Is it clear? So I repeat the question. Epithelium plus lamina propria is called?
Mucosa.
Mucosa. This is very important. Again, Professor Antal question. You can point here. This is your future oral question right here. What is mucosa? Epithelium plus lamina propria. Okay. Somebody has said something. I heard it. Lymphoreticular connective tissue. I heard it. Somebody have said it when we were talking about what is lamina propria. Lymphoreticular connective tissue. What does this mean? Lymphoreticular connective tissue. What does it mean?
The connective tissue has a lot of reticular fibers.
Perfect. This connective tissue has a lot of reticular fibers. Reticular fiber is which collagen?
Type 3.
3. This is a collagen type 3. Which connective tissue has lots of reticular fibers? Loose connective tissue. So you remember from last semester, we have dense connective tissue and we have loose connective tissue. Loose connective tissue is filled with reticular fibers. So lymphoreticular connective tissue, this is a perfect description of lamina propria. If they ask you, what is lamina propria, right? They ask you the stupid question, like, how does it look like? Your answer will be lamina propria is lymphoreticular connective tissue. If they ask you, what does it mean, lymphoreticular connective tissue? You will say, well, lympho, it has lymphocytes. Reticular, it has lots of reticular fibers. Which kind of connective tissue has lots of reticular fibers? Loose connective tissue. So what is lamina propria? It is a loose connective tissue filled with lymphocytes. Am I clear? Yes, no?
Yes.
Yes.
Yes. Okay. So back to the mucosa. So we have said that this is our oral cavity, and within the oral cavity, we have mucosa. Everywhere in my oral cavity, there is mucosa. There are 3 types of mucosa: lining, masticatory, and specialized mucosa. Okay, so within one oral cavity, you'll find 3 different types of mucosa: lining, masticatory, and specialized. Any guesses where would you find lining exactly in the oral cavity? Like which structures in the oral cavity will be lined by lining mucosa? Think.
Stomach, intestines.
In the oral cavity.
Palate.
Forget the rest of the GI. Which structures in the oral cavity will be lined by lining mucosa?
Palate.
Depends which one, hard or
Hard.
soft?
Hard.
Soft.
Okay, so soft palate will be lined by the lining mucosa. Okay, anything else?
The salivary gland.
Salivary glands are a bit hidden, so they will not be lined by anything.
The tongue.
The tongue is a very special structure in our oral cavity. What is so special about it?
It has taste buds.
Very good. So that's why it is lined by specialized mucosa. Tongue is the only structure in our oral cavity which is lined by the specialized mucosa. I repeat, what is so special about the tongue? It has taste buds. It's capable of tasting, so not. Lining, soft palate, and 2 more. Think about it. Lines.
Cheek.
Sorry?
Cheeks. The inner cheek.
Sorry?
Upper.
Inner cheeks, very good. So the inner part of the cheek, if you open it up right now, I'm so sorry, but it's actually lined by the lining mucosa. And also, what is this? The inner part of the lip. This is also lining mucosa, all of this. The inner component of the lip, this is also going to be lining mucosa. So lining mucosa lines the inner part of the cheek, the inner part of the lip, and soft palate. Specialized mucosa will be covering the tongue. What is so special about the tongue? Taste buds. So that's why specialized mucosa contains taste buds. Masticatory. What does it mean, mastication?
Chew.
Chewing. Means chewing. It's the action of chewing, right? The process of chewing. So which structures in the oral cavity has to chew except for teeth?
Tongue.
Yes, but
The,
sorry?
the gingiva.
Gingiva, perfect. Masticatory is gingiva. So your gum or gingiva, if I open it up for you right now, I'm so sorry. Do you see this red part? This is your masticatory mucosa. So gingiva is masticatory mucosa. They're interchangeable because it's quite visible right there. So gingiva, yes. And think about it. Tongue does participate in chewing, but not as you think it would. Basically what it does, it pushes the food against something. What do we push the food against?
Hard palate.
Hard palate. So this is why, what is the another thing which is covered by masticatory mucosa? This is going to be hard palate. So gingiva and hard palate, they're covered by masticatory mucosa. Is it clear until this point?
Yes.
Okay. So
Yeah.
let's go more details. Lining mucosa. So we have said covers the inner surface of the lips, cheeks, and soft palate. What is mucosa? 2 layers. What are those 2 layers?
Epithelium.
Human.
And lamina propria. What is so special about lamina propria?
Has lymphocytes.
It has lymphocytes. Okay, so this particular connective tissue will have lots and lots of lymphocytes. Now, epithelium. Which kind of epithelium will be present in the lining mucosa? As you can see, stratified squamous, non-keratinized epithelium. How many layers? Fun fact. Majority of people fail not because of histo 2, but because of histo 1, because they forgot the histo 1, because they spent entire semester preparing for histo 2 and completely neglected the histo 1 part. Lots of questions, especially in the oral, will be coming from histo 1, lots of them. Okay, so this is why with me, every little test before the class begins, we will always have histo 1 questions, so you can keep it fresh. There will be 3 layers in the non-keratinized stratified squamous epithelium. Starting from the bottom, which one?
Stratum
How do you
basale.
call it? Basale, stratum basale. Okay, stratum basale is the most bottom one. Next one?
Polygonal.
Very good. So in everywhere in the world, if you come out of Hungary, you will call it stratum spinosum. But in our university, this department decided that it's actually going to be called stratum polygonale. So in Hungary, University of Debrecen, the second layer is polygonale. I repeat, go out of Hungary, go to Austria, tell them this is stratum polygonale. They'll be like, was? You know, they will not understand what are you talking about. So because they have stratum spinosum there. So second is stratum polygonale. And then the last one, everywhere in the world, is called stratum superficiale. But in our department, it's called?
Panocellulare.
Very good. It's called plan cellulare. Okay, so the last one is called plan cellulare. So 3 layers, basale, polygonale, plan cellulare. How do you know that the epithelium is non-keratinized?
The uppermost layer of the cells have nucleus.
Perfect. Do not forget the following situation. What does it mean keratinized? Keratinized means it's dead. It's filled with keratin. The epithelium, the most superficial epithelium, is dead. How do you know that those cells are dead? They do not have nuclei. That's it, they're dead. So if the epithelium is non-keratinized, what does that tell you? It tells you that it's still alive. So the most superficial cells in the plan cellulare will have their nuclei and will not have keratin. So I repeat, how do you know that you're looking at the non-keratinized epithelium? The most superficial layer, plan cellulare, the cells there are alive. How do you know that they're alive? They have the nucleus, but they do not have keratin. Speaking of keratin, famous question, what is keratin?
Dead cells. Without
Dead cells have it, but what is keratin? About 80% of the students say it is a
class.
Wait. They say it is a protein. Okay, so about 80% of the students, what is keratin? It is a protein. And you're absolutely right. Okay, so keratin is a protein. But do not forget that we're in histology. And in histo, everything is classified. Everything undergoes classification. This belongs to that belongs to this, and etc. Okay, so keratin is a protein that belongs to which group of proteins? How do we call those proteins? Somebody has started talking about it? No? Intermediate filaments. Keratin is an intermediate filament. Can you tell me what are the 3 components of cytoskeleton? Everybody knows what is a cytoskeleton?
Microtubules, intermediate filaments, and actin filaments.
Perfect. The three components of cytoskeleton are microtubules, actin filaments, and intermediate filaments. Cytoskeleton, it's in the name. It is a skeleton of a cell. This is the thing that makes up the cell. What is the job of microtubules?
To absorption.
Microtubules. You remember from last semester, we have learned that microtubules, they assemble and disassemble, assemble and disassemble. They're not a constant structure in a cell. They're not a rigid structure. They constantly assemble and disassemble. So what do we use microtubules for?
Spindle formation during cell division.
Sure, that's so specific, but generally, microtubules, what do we use them for? What is it good for? Assemble, disassemble, yes?
Transport from one
Transport.
part to another.
Transport. Microtubules equals transport. Microtubules are always used for transport. The spindle formation is very, very, very specific. It is good, but it is like little specific part. Generally, in a cell, microtubules are used for transport. That's why it's important that they're not a constant structure. They're not a rigid structure. I assemble it, send something along that microtubule, and then disassemble, and then assemble again, send something somewhere else, and then disassemble, and etc. Okay, actin filaments, what can they do? Actin filaments, come on guys.
Movement.
What can actin do?
Contraction.
Sorry?
Contraction.
Very
Contraction.
good. They contract. So what good is it for a cell? A cell can move for movement. If actin filaments can contract, think cilia. You remember cilia? They had 9+2 arrangement, right? So there we had microtubules, but there were also a little bit of the actin filaments also present, like microvilli or something like this, stereocilia, and etc. So actin is definitely for movement because they can contract and relax and etc. Now, intermediate filament, last thing, which keratin is a part of. So don't forget, we're still talking about keratin, but it kind of brought us there, okay? So we are refreshing our memory. What do intermediate filaments do? What is their most important function? One word, that's it. Shape. Intermediate filaments are responsible for a shape of a cell. They are responsible for a structure, morphology of a cell, all right? So if you compare intermediate filaments with actin and microtubules, you will notice that intermediate filaments are rigid structures. They're constant structures, right? Microtubules assemble, disassemble. Actin, contract, relax. Intermediate filaments are always stationary. They're always like, I don't know, stable. They don't move, they don't change. So once I put it there, that's it. So why would we want to put keratin in a dead cell, right? Because think about it. Keratinized epithelium is dead epithelium, right? So why would I want to put keratin in a dead cell? Think about it as taxidermy. Do you guys know what is taxidermy? Yes, no? No?
Mm-hmm.
This is a very, very weird hobby, extremely weird. Whoever has this as a hobby needs to go and check themselves out. Basically, you kill an animal, you remove its like organs, and then you stuff it with something and make a statue out of it. So, do you know what I mean? Taxidermy, have you ever seen it? No, there's like, I don't know, moose or like, I don't know, wolf. Some people, especially in like, I don't know, forests,
stuff their cats
houses.
and stuff.
Exactly. Okay, so they, I don't know, they kill the animal so they can preserve it. Okay, so they remove the inner part, so they stuff it with something, so it still looks like it, but it is no more alive. The same concept is here. Keratinized epithelium, your skin right here. This is keratinized epithelium. What's the concept? The most superficial cells are dead. but you want to stuff them with something hard so they still preserve the shape. What is that hard? It's the keratin. Keratin is the intermediate filament, which is responsible for kind of holding, preserving the shape of a structure. Is it clear?
Yes.
So if Professor Antal asks you this stupid question, why would you want to put keratin into a dead cell? What's your answer?
Preserve the shape of the structure.
Exactly. So keratin is an intermediate filament, which is a part of a cytoskeleton. And intermediate filaments are rigid structures. They are known to preserve a shape. So if I have a dead cell, which I don't want to get rid of, I would rather stuff it with something hard, such as keratin. That would be your answer, okay? Now, I think it's clear, right? So non-keratinized epithelium is present within lining mucosa, 3 layers, plant cellulari, has the nucleus, does not have the keratin, etc. Lamina propria is connective tissue, this loose connective tissue with lymphocytes, but you consider it is very special because this particular connective tissue has more elastic fibers and less collagen fibers. This is why I can do this, right? So I can stretch my lip, I can stretch my, I don't know, cheek, I can do anything I want because the lamina propria there possesses more elastic fibers. Remember this, they will ask you this in the SCT. They will ask you to compare the mucosa's lamina propria in the lining, masticatory, and specialized, okay? So specifically in the lining mucosa, lamina propria here has more elastic fibers. Now, how do we call this?
Mucosa.
Mucosa. Whatever is under mucosa is called
Submucosa.
submucosa. This is very important, very important. Lining mucosa is the only one to have submucosa under it, the only one. So if they ask you this question, what does lining mucosa attaches to? It attaches to the submucosa, okay? Masticatory mucosa, what does it cover? What does masticatory mucosa cover?
Your gingiva and your hard palate.
It covers the gingiva and the hard palate. So if there is no submucosa, what will masticatory mucosa attach to? What's under?
Basal lamina.
Sorry? Could you repeat?
Basal lamina.
What is, we have said this is the gingiva. What is right under it?
It's a bone.
Bone. So what does masticatory mucosa attaches directly to?
Bone.
The bone. Okay, so there is no submucosa. So masticatory mucosa will attach directly to the bone. Yes. Specialized mucosa is in the tongue. There's also no submucosa. So what will it attach to?
Muscles.
Muscle. To the skeletal muscle of the tongue. So I repeat, the lining mucosa is the only one which attaches to the
Submucosa.
submucosa. So submucosa is only present under the lining mucosa, okay? So it's only present under the lining mucosa. The masticatory and specialized mucosa do not attach to it because it's not there. So what will the masticatory mucosa attach to?
Bone.
Bone. What will the specialized attach to?
Skeletal muscle of tongue.
To the muscle of the tongue, which is the skeletal muscle. So what is it, the submucosa? It is connective tissue, and this connective tissue has lots and lots of salivary glands. Okay, so submucosal salivary glands. Okay, I will get back to it. So masticatory mucosa. We have said gingival hard palate. Again, how do we call this thing together?
Mucosa.
Mucosa consists of epithelium and lamina propria. Notice, the lamina propria of masticatory mucosa has lesser elastic fibers and more collagen. So can you, I don't know, stretch your gingiva? You cannot. So why? Because there aren't that many elastic fibers. Now, how's the epithelium in the masticatory mucosa? It is keratinized epithelium. So how many layers? My favorite question. How many layers?
4.
So, okay, let's do 4. So what are the layers from the bottom?
Stratum basale.
Basale, then
Spinosum.
spinosum, very good. Stratum spinosum, then
Superficiale.
Granulosum.
granulosum. Fourth, the last one.
Fasciale.
What with lucidum?
Corneum.
I heard 2, lucidum, corneum. So corneum is definitely there. So let's put it there, corneum. So stratum corneum. But where is lucidum?
In thick skin.
In thick skin. Where do we have thick skin?
In palms of the
No.
hand. And
The
shoes.
palms and the soles, right? So palms and soles, they will have the thick skin. Okay, lucidum, is it a separate or is it is it a separate layer or is it a sub layer? And if it is a sub layer, it is a sub layer of what?
It's a sublayer of granulosum.
Not of granulosum, but it is a sub layer of
Corneum.
corneum. Okay, so this is why you have to remember, if they ask you this question, how many layers is keratinized epithelium? Your answer is, that depends. Are we talking about thick skin or are we talking about thin skin? In the thick skin, how many layers there are?
Why?
Kind of. So there are 5 layers, but lucidum is not technically a separate layer. It is a sub-layer of
Corneum.
Corneum.
corneum. So total layers are 5, but lucidum is not a separate layer. Do you understand what I'm trying to say?
Yeah.
It is a sub-layer. So if I were to count, yes, there are 5, but technically there are 4 because lucidum is kind of part of a corneum. Yes?
Yeah.
Yeah.
Thin skin, does it have lucidum?
No.
No.
No. So can we quickly do a comparison, thin and thick skin? What are the major differences? So number 1.
The lucidum. The stratum lucidum.
Very good. Thick has lucidum, thin doesn't have lucidum. Next.
Hair
Hair
follicle
follicles.
Thin,
has hair.
Yes.
it has, so what did I write here? It doesn't have, sorry. So thin has the hair follicles, thick
Doesn't have.
doesn't have the hair follicles. And last, why do we call it thick skin?
Oh, the corneum is thick.
Exactly. The corneum is triple the thickness. We call it thick skin because corneum is extremely thick. Okay, so corneum is thick. While in the thin skin, the corneum is normal. It's like this much. That's it. So those are the 3 differences between the thin and the thick skin. Is it clear?
Yeah.
Okay, so now let's kind of compare. We have non-keratinized epithelium and we have keratinized epithelium. So non-keratinized is in the lining mucosa. Keratinized is in the masticatory mucosa. So how are the nuclei? Do non-keratinized cells have nuclei?
Yes.
Yes.
Yes. Keratinized?
No.
No. Keratin. Is it present in non-keratinized?
No.
No. Okay, so it's not there, and in keratinized it is. But we have a 3rd division, which is called para-keratinized epithelium. Okay, so para. It is right here in the middle. How will it look like? What will it have? It is in the middle. So
No nucleus, but there's keratin. Oh, Yeah.
But then it is keratinized.
There's both
Has
nucleus
both.
and keratin.
Has both. It has nuclei and it has keratin. So remember this, okay? So parakeratinized epithelium is very, very special because it has nuclei and it has keratin. The nuclei which are present in the parakeratinized epithelium are pycnotic. This is very important SCT question, okay? So they will ask you about this in the oral as well. So which kind of nuclei are present in the parakeratinized epithelium? You see the two P's, right? P and P. So pycnotic nuclei are present within parakeratinized epithelium. What does it mean, pycnotic? It means highly condensed. So if I were to draw a normal cell with a normal nucleus, this is how it looks like. How will I draw a pycnotic nucleus? So here is a cell, and pycnotic nucleus is a little condensed dot. That's the difference between normal nucleus and pycnotic nucleus, okay? Now, where will I have this parakeratinized epithelium? I will have it everywhere in the oral cavity. So what do I mean? From protection perspective, right? So protection perspective, which epithelium is better, keratinized or non-keratinized?
Keratinized.
Keratinized, because it has keratin. This is a hard substance. It's like an armor that fully protects the cells behind, right? In your oral cavity, do you need protection or not so much?
You need protection.
You need protection?
Protection. I don't know. I take a bite of an apple. I start chewing it. This is a hard substance, right? Hard surface. So it's constantly exerting pressure everywhere in my oral cavity. So I would rather have keratinized epithelium, but my lining mucosa, is it non-keratinized or keratinized?
Non-keratinized.
Keratinized.
Non-keratinized. Let's draw it. This is my non-keratinized mucosa, a simple line. The first layer is epithelium, under it lamina propria. Which kind of epithelium is in the which kind of epithelium is in the lining mucosa? Non-keratinized. This. I took a bite of an apple. This apple, I don't know, cut the surface here, right? So what's going to happen here? You want to protect yourself somehow. So you're going to put keratin. You're going to put keratin here. Which kind of epithelium is here?
Non-keratinized.
But it has keratin.
So parakeratinized?
Parakeratinized. Why para and not keratinized?
Because nucleus is still present.
Because the nucleus is still present. Because nucleus is still there. For those who did not understand, let's draw it again. So epithelium is here. This is lamina propria in my lining mucosa. Am I right? So here we have lots and lots of cells. What is the shape of those cells? They are stratified squamous, non-keratinized. Am I right?
Mhm.
Yes. Some kind of injury occurs. When you were chewing, I don't know, you were doing something with your oral cavity, there is lots of pressure in this area, let's say, this area specifically. Not here, in this area. You want to protect yourself from that injury. What would you want to put in those cells? You want to put keratin here. There's keratin here. So those cells have nuclei and right now they have keratin. So which kind of epithelium is this?
Parakeratinized.
Parakeratinized, because it has the nuclei and it has the keratin. Which kind of epithelium is this?
Oh, non-keratinized.
Non-keratinized. This one?
Non-keratinized.
Non-keratinized. Do you get it?
Yes.
Okay, continue. We continue. This is still non-keratinized epithelium along my cheek. Still, non-keratinized epithelium. Again, I was eating something damaged. Boom, here. What do you want to do?
Put keratin.
Put keratin. I put keratin. How would you call this epithelium now?
Parakeratinized.
Oh,
Parakeratinized. But this everywhere else is
Non-keratinized.
Non-keratinized.
Non-keratinized. So a question for you. Lining mucosa consists of only non-keratinized epithelium, correct?
False.
False, because you can see that there are sometimes patches of parakeratinized epithelium present. What do those patches represent? They represent that that area was under some kind of stress. There was some kind of injury happened there. Is it clear?
Yes.
So once you see parakeratinized epithelium, it's okay. It just tells you that that particular area has experienced some kind of force or damage or injury or stress or something like that. Am I clear?
I have a
You need to
question.
understand that parakeratinized epithelium is present everywhere in the oral cavity. It doesn't matter if you're talking about lining, if you're talking about masticatory or specialized. Each of their epithelium can become parakeratinized. Am I clear?
Julia,
Yeah.
can parakeratinized become non-keratinized again?
Not really. Once you put the keratin, there's nowhere else for it to go. There's no enzyme that can remove the keratin. So parakeratinized can become keratinized. And keratinized will eventually just peel off and just die, but it cannot return to the point. Okay, so non-keratinized can become para. Para can become keratinized, but you cannot return to the point. Am I clear?
So if parakeratinized is damaged, it become keratinized.
Yes. Let's imagine I keep on eating that apple. I keep on eating, right? I keep on putting more and more damage. Eventually, chronically, para can become keratinized, yes. So, any questions?
I have a question for specialized mucosa.
Yeah. We haven't talked about it yet, so yeah.
Okay.
So, what's the question?
Okay, because we had discussed earlier that it has no submucosa, right?
Yeah.
So what is the specialized mucosa attached to? What part of the tongue?
To the muscle. So tongue itself is a muscle. It's a skeletal muscle. So the superficial specialized mucosa attaches to the muscle of a tongue, the superficial part of a muscle of a tongue. In the masticatory, it attaches to the bone, superficial part of the bone of a jaw, for example, right?
Thank you.
So specialized mucosa tongue, what is so special? What's special about specialized mucosa?
The taste buds.
Presence of the taste buds. Now, if I draw my lining mucosa, I draw it smooth, very like a line. By the way, in the oral, Professor Rosa, Professor Clara, if you pick them, brace yourself because you're about to draw things, okay? So you are going to be drawing something. So try for anything that we're learning, you can see that I'm always drawing something. Try to remember those drawings. They will help you in your oral exam to get a 5 and not like a 3, okay? So make sure that you remember. So if you draw a lining mucosa, it's just a line. If I draw a masticatory mucosa, it's also just a line. But if I draw a specialized mucosa is not smooth. Take a look at your tongue. It has lots of bumps, am I right? It has lots of little tiny protrusions. So this is why you have to remember that if you draw specialized mucosa, you draw it like this. You draw it with protrusions of some sort. How do we call those protrusions? They're called papilla. Okay, so specialized mucosa consists of lots and lots of weird looking protrusions. So it's not smooth like masticatory or lining mucosa. It's not smooth. It has lots of protrusions. Those protrusions are called papilla. What is so special about papilla? They have taste buds. So within the papilla, you have taste buds. So if I ask you, where are the taste buds? Specialized mucosa. But where exactly in the papilla of the specialized mucosa? Does it make sense?
Yes.
Okay. Finally, let's start our first slide. Okay, we'll start with the lip. and then we'll return to the specialized mucosa. So lip. Here's the anatomical kind of histological looking lip. Lip has 3 zones. How do you call the most inner part? We talked about it today. The inner part of the lip and cheek, how do we call it?
Lining mucosa.
Lining mucosa. Okay, so the inner part of the lip and cheek, the most inner part. So until here. Okay, so this is going to be the lining mucosa, the inner part of the lip. The red zone, right? So this area where I put the lipstick, this is known as the vermilion or simply red. Okay, so this is the red zone. So this is the visible part where I put the lipstick or whatever. And the third zone of the lip is the skin. So it is right under the lip. Okay, so right under the lip, whatever little part of the skin we have here. So this is the skin. In anatomy department, they call it the cutaneous segment. Okay, so this is going to be known as the cutaneous segment. Cutaneous segment. So lip has 3 zones. The inner one, it's called
Lining mucosa.
lining mucosa. This one, the visible one,
Vermilion.
vermilion zone or red zone, and then right under it, somewhere here,
The thin skin.
skin or cutaneous segment. Here, that's what they did last semester. They pointed last year, sorry, they pointed here and they ask you, what is this? This area right here between the red zone and the skin, this is known as the vermilion border. Okay, so this is the visible border of the lips. This is where you put the lip liner, but right now people overline their lips, right, to make it bigger. So the vermilion border becomes higher than it naturally is. But this is going to be known as the vermilion border right here. Okay, 3 zones of the lips. What is in the core of the lip? What is inside of the lips?
Muscle.
Muscle. Which kind of muscle? Skeletal, smooth, cardiac?
Skeletal.
Skeletal. How do you know it's skeletal? How do you know? Without even looking at it, without anything, just with using your brain, how do you know that the muscle, which is in the lip, is skeletal? What's your answer? I can do this. I'm talking right now, so
I can move my leg, the muscle there.
I am in control. This is a voluntary muscle. Skeletal muscle is a voluntary muscle. You are in control. Okay, so you can initiate or stop the movement. Smooth and cardiac, they're involuntary muscles. You're not in control whatsoever. Your higher brain powers are in control of that, but not you, definitely. Okay. How do we call the muscle which is inside of your lip? We call it orbicularis oris muscle. Okay, so you will learn it in the anatomy. It's like a circle. Okay, so it's called orbicularis oris muscle. This is a skeletal muscle which is in the core. Now, what could be the innervation to this muscle? This semester, you're going to see that I'm going to be obsessed with innervation. This is just to help you for your anatomy. For every structure, I'm going to be asking you the nerve because that's what they do anyway. So what could be the nerve for the facial muscle, orbicularis oris muscle? What could be the nerve? Facial muscle. Facial nerve, okay? So facial nerve. This is a cranial nerve, number?
7.
7. Okay, so this is cranial nerve number 7. Okay, so let's take a look. This is our slide right here. So what can we see? Now, immediately when you can take a look at it, you see that we have 3 zones. So this is the first zone that begins, follow, follow, follow. It looks the same, finishes here. This is the second zone, finishes here, and this is the third zone, right? So we have 3 zones, just as the lip should. The stenion is obviously H&E. Without magnifying, what's the epithelium in this zone? What's the epithelium here? Come on, guys. Is it lots of cells or little?
Little.
No, it's lots of cells. Oof, 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 oof. There's so many cells here, like 20 layers. So is it simple or stratified?
Stratified.
Stratified. With this magnification, I can tell you they're squamous. Okay, I can see it already. Okay, so they're going to be squamous cells. So this is a stratified squamous epithelium. We have two types, non-keratinized or keratinized. So can I tell you from this magnification?
Keratinized.
So can I tell you or I cannot tell you? Is it keratinized or non-keratinized? From this magnification, can I tell you?
Yes, you can.
Yeah.
So what do I need to look for?
The keratin layer.
Very good. How does the keratin looks like? Is it darker or paler?
Pale.
Darker. Keratin is
Oh,
extremely,
okay.
extremely dark band. Okay, so on our slide, it will be extremely, extremely dark band. Do you see a dark band here?
No.
No. So what is the full name for this epithelium?
Non-keratinized, non-keratinized.
This is going to be stratified squamous, non-keratinized epithelium. I repeat, I don't even need to magnify. From this part, I can tell you everything I need to know. Again, keratin is dark, okay? It is a dark band. It's not there. Take a look at the most superficial layer. It's definitely not there, okay? So this is a stratified squamous, non-keratinized epithelium. Which kind of structure in the lip has it? Which zone has non-keratinized epithelium?
The first one, the lining mucosa.
Lining mucosa. So from here until here, how would you call this segment?
Lining mucosa.
Lining mucosa. This is the epithelium of the lining mucosa, and under it is the lamina propria of the lining mucosa. Which layer is here, which is under?
Submucosa.
Submucosa, very good. Do not forget that your lining mucosa is the only one which attaches to the submucosa. As you can see within the submucosa, we can see some structures. What are those structures?
The taste.
Glands.
Oh, the glands.
Glands. Which kind of glands?
Salivary glands.
Salivary glands. We have three types of glands, mucous, serous, and mixed. Which one is this?
Serous.
Mixed.
We learned it last semester. So if it is serous, it will be dark, like those ones. Those are the dark ones. But take a look. You also have pale acini. So
Mixed.
mixed. Okay, so those salivary glands, they are going to be mixed submucosal salivary glands. They are in the lip. So how would you call them? What's the Latin name for lip?
Labia.
Again?
Labia.
Very good, labia. Okay, so what will be the full name for this? This will be the labial submucosal mixed salivary glands. Yes. Now, okay, this is the next zone. And the first thing that you know, yeah, so labial submucosal mixed salivary glands. That's the full name for them here. So in the next, yeah?
Oh, thanks. I was just saying thank you.
Ah, okay, okay. No need to think. So yeah, I just keep on going. So you can see here the dark band immediately. So what is this dark band?
Keratin.
Keratin. So how's the epithelium here?
Stratified squamous keratinized epithelium.
Very good. This is a stratified squamous keratinized epithelium. Which zone is next to the lining mucosa? We have said this is a lining mucosa. Which zone is next to it?
Vermilion.
Vermilion. So all of this, which has stratified squamous keratinized epithelium, this is going to be vermilion zone. So this zone right here, this is a red or a vermilion zone, which is visible everywhere. So right next to it is this zone. Which zone is this? What's under?
Skin. The thin skin.
The thin skin. How do you know this is a thin skin?
Hair follicle.
Hair follicles. So those weird looking structures, those are the hair follicles. As the hair follicles are present, okay, so you have to understand that this is the thin skin. Okay, so mucous segment or lining mucosa is this. This is the vermilion zone or red zone, and this is the cutaneous segment. Those structures here, those are the labial, submucosal, mixed salivary glands. Now, in the core of the lip, you can see this structure. It's kind of in the center of it, right? So this one, what is this? What is it?
Orbicularis.
Orbicularis.
This is going to be the orbicularis oris muscle. This is a skeletal muscle. This is the orbicularis oris. What is the innervation?
Facial muscle.
Facial nerve. Facial nerve. So we magnify each segment a little bit. So this is the lining mucosa or mucosal segment. How's the epithelium? What is the epithelium?
Stratified squamous non-keratinized.
Very good. What is this layer?
Stratum basale.
Very good. Until here?
Stratum spinosum.
I cross it out. It's not spinosum. In our university, it is
Polygonale.
polygonale. Okay, so added here. Apologies. Okay, so this is going to be the stratum polygonale, and the most superficial.
Planocellular.
Plant cellulare. This is, as you have said, non-keratinized epithelium. How do you know?
No keratin.
No keratin, and
Present nucleus.
it has the nuclei, right? So nuclei here, those are nucleus, this, all of them have nuclei. So this is a non-keratinized epithelium. What is this? Like this. What is under the epithelium?
Oh, lamina propria.
Lamina propria. This is the loose connective tissue. What are those cells inside of it?
Lymphocytes.
Lymphocytes. So those dots that you can see here, those are going to be the lymphocytes. Those basophilic nuclei, those are going to be the lymphocytes. Under it is just submucosa, which is the loose connective tissue. Okay, so this is the loose connective tissue, submucosa. Any questions? So we have said in the submucosa, we have the salivary glands. So what is the full name for this gland?
Labial mixed submucosal salivary gland.
So labial submucosal mixed salivary gland. What is this? If I point here and ask you, what is this? We learned it last semester, so you have to tell me the precise name. What is it?
Epimeloom.
Mucus
And there's the same here. Mucus acinus, right? So if it is one, it is acinus. What is acinus? Do you remember?
A secretory unit.
Very good. This is a secretory unit of a salivary gland. It usually looks like a sac, like this, right? So this is going to be the acinus. And from acinus, we have duct. So this is a duct, and this is a duct, and this is also a duct, right? So acinus, and then something that comes out of it is going to be a duct. Inside of the acinus, we have epithelial cells, and those epithelial cells make the saliva, for example, right? So they make either mucus or serous saliva. How do they put the saliva into the duct? What surrounds the acini? Here, like a flat cell. What surrounds it?
The cell.
The cell. Which cell?
Yeah.
Myoepithelial cells. Okay, so myoepithelial cells are going to be surrounding the acinus. Myoepithelial cells will be contracting, and as they contract, they kind of push all of that saliva into the duct. Okay, so how does the how does the saliva end up in the duct? Because of the myoepithelial cells. As you can see, the mucous acini are paler, right? They're much paler than the serous acini. Why? Why the mucous acini are paler?
Because they produce decreasing-rich in carbohydrates.
So one by one,
The mucus
somebody?
is washed off during fixation.
Very good. Mucus is washed out. During which step?
Fixation.
Fixation is good, but I want another step.
Formula.
Clearing,
That's fixation.
clearing,
Clearing. Clearing with what exactly?
xylitol.
Xylo. Okay, so this is a favorite question of Professor Rosa. She will always ask you this. From last semester, remember, in first histo ACT. So we have lots of steps in preparation, but the mucus specifically is lost during fixation and during clearing. Clearing with organic alcohol, which is going to be known as the xylol. Okay, so xylol. Xylol is organic alcohol that will be washing out the mucus here. What if I wanted to see the mucus? What would I stain it with?
Yes.
PAS, right? So PAS in Hungary, everywhere else in the world, it's PAS. What does PAS stand for?
Periodic acid.
Periodic acid.
Schiff base.
Base.
What would be the color?
Magenta.
Purple.
Yeah.
Magenta.
Magenta. This is a very important difference. Hematoxylin, purple. PAS, magenta. Even though they're on the same shade, it's like purplish color, but the color for PAS is going to be magenta. Okay, which acini are those?
Serous.
Serous?
Serous. What is the color for serous acini? We say it is basophilic, but why? And you remember those students who I have taught last semester, there's a specific way to answer that question. How do you describe it? Why is this acinus basophilic?
Does it produce secretion-rich in proteins?
Perfect. Because it makes lots and lots of proteins. Not because it contains proteins, but because it makes proteins. Which organelles do I need to make a protein?
Rough and
RER.
So let's start from the beginning. What is the first thing?
Ribosomes.
Nucleus. Nucleus, DNA, okay? Then?
RNA.
So where is the RNA? Ribosomes and RER, okay? So nucleus, a rough endoplasmic reticulum, and ribosomes, those organelles are responsible for making a protein. What is their charge?
Of proteins?
Negative.
What is the charge of the nucleus, RER, and ribosomes? What is their charge?
Negative.
Negative. So this is why a cell is basophilic. It likes the basic things. Because base has which charge?
Positive.
Positive. If a cell contains lots of proteins, what will be its color?
Red.
Eosinophilic. Because protein has which charge?
Positive.
Positive. And eosin likes that. Do you see there are lots and lots of red things? What are those red things?
Collagen.
Red lines. What are they?
Collagen.
Collagen. What is collagen? What is it? It is a protein. So do you see? It is red. So collagen is red. Is it clear? They also like to ask this to explain the basophilia and eosinophilia. Do you remember this? So was it clear right now? A little bit? Okay. Okay, I point, you tell me, what is this?
Serous acini.
Serous acini.
The dog.
Now this is serous acinus, and this is a serous acinus,
Oh,
and this
the
is
duct?
also serous
The
acinus.
duct.
This is a duct. This is a duct. This is a duct. Which cell is this?
Myoepithelium.
Myoepithelial cells. Very good. Okay, so this is a perfect example of how it should look like. So on the top of the acinus, you'll always find myoepithelial cells. Okay, I forgot to ask you a question here. What is the major difference between exocrine and endocrine glands? Famous question of every professor. Pretty much every professor is going to ask you this question in the oral exam. What is the difference between exocrine and endocrine glands?
The presence of the endocrine glands don't have a duct.
I heard it. Exocrine have duct. Exocrine glands have a duct system. This is a way to answer, okay? The major difference between exocrine and endocrine glands is that exocrine glands possess a duct system, while endocrine do not have a duct system. Where do they drop their secretions? Directly into the blood. This semester, we're going to see that all of the endocrine cells surround blood vessels always, because they need to directly put their secretion into blood. Okay. So I want you to ask something. I think I've seen it here. Wait just a sec. I feel like I'm forgetting something. Where is it? Where is it? Okay, it's not there. Okay, later. So, cutaneous, how do you know that this is a skin segment?
There's hair follicle.
Hair follicle.
Hair follicles. The first thing that helps you, hair follicle. Once I see it, that's it. How's the epithelium?
Keratinized.
Keratinized. Which layer? Let's try to do the layers. The first layer?
Stratum basale.
Basale. Next one?
Polygonale.
This is keratinized epithelium, so
Granulosum.
spinosum, guys. Okay, so do you see the dark line? What is this? Granulosum. Okay, so the next layer which has the dark lines is the granulosum, and the most superficial is
Corneum.
corneum. Okay, as you can see, lucidum question mark. Why is it not here?
It's thin skin.
It is a thin skin. What are those?
The sebaceous gland.
Very good. Those are the sebaceous glands. What is the type of secretion for sebaceous gland?
Holocrine.
Holocrine. Okay, remember this, that the way sebaceous glands make things, they make it via holocrine type of secretion. Can you describe it to me?
So when they are secreted, the whole cell is dies along with their secretion.
Very good. Apoptosis. Okay, so one word, it's going to be apoptosis. Holocrine secretion means that the sebaceous cell, let's magnify it a little bit, right? So the sebaceous cells, here they're normal looking cells, but do you see something happens to them? What do they lose?
Nucleus.
Nucleus. This is a form of cell death. When you lose the nucleus, this is the apoptosis. So the way that the holocrine secretion works is that the sebaceous cells, they have to kill themselves. You can clearly see how they're killing themselves here by slowly getting rid of the nucleus, which is going to be present here, but it is absent here. Okay? What is this?
The hair.
Hair follicle. Okay, so this is a hair follicle. Hair will have keratin, which will be visible. Here, there is no keratin. Now, this is very important part that they always test. Sebaceous glands, are they exocrine or endocrine glands?
Exocrine.
Exocrine. They're exocrine glands, but you have to remember that sebaceous gland, even though it is an exocrine gland, it lacks ducts. Sebaceous glands are the only glands, which are exocrine, that lack the duct. Am I clear? So those things which you can see here, those are not ducts of the sebaceous glands. Those are dead apoptotic, dead underwent apoptotic cell apoptotic process cells. Okay, so they're dead here. Those are not ducts. Sebaceous glands are the only glands which lack the ducts, yet they are exocrine. The only sebaceous gland that has a duct is where your eyelids are. Okay, so the eyelids. There we have a Meibomian gland. It's not important. It's next semester neurohistology. And there it has a duct. Do you know what is sty? Have you heard this word before? No? It's like a pus on the eyelid. Have you ever seen it? Like sometimes people have it. Okay, there's like a pus on the eyelid. This pus is going to be inflammation of the duct of the sebaceous gland in the eyelid. Okay, so that's the only place where we have the duct in the sebaceous glands. Everywhere else in our body, the sebaceous glands do not have it. Clear? Transition is more or less abrupt. Okay, so in the SAT, they will give you the option that transition between zones is smooth. That's not true. It's abrupt. You can see that every zone ends very, very abruptly. Okay, just suddenly it ends and another zone begins. There is no smooth transition between the zones. Okay, red zone. So this is, what is another name for the red zone?
Vermilion.
Vermilion. Okay, so another name, this is known as the vermilion zone. So how do you know that this is a red zone? You know, thanks to those protrusions here. Those protrusions are called papilla. Do not confuse these papilla with the specialized mucosa of the tongue papilla. They're not the same thing. Okay, so you remember papilla? It was the protrusions on the tongue. It was the protrusions of the specialized mucosa. Here, it's something else. So, can you tell me which kind of epithelium is this? Vermilion zone. Which epithelium? Sorry?
Stratified squamous keratinized.
Very good. It is stratified squamous keratinized epithelium. Okay, it is keratinized. What about here? And here? And there? Is it keratinized? And here?
Yes.
What is this dot? Do you see the dot?
Yes.
Okay, so what could this dot be?
So it's keratinized. Oh, so it's non-keratinized.
No. It is keratinized because you can see granulosum, spinosum. It is parakeratinized. So those areas right here, those are the parakeratinized epithelium. It's very, very evident. So it has the keratin, it's very dark, but it also has a darker dot. And these darker dots are the nuclei. Those are the nuclei of the cells. Okay, so little tiny dots, which you can see here, those are going to be the here as well, for example. And here, okay, so it has lots and lots of nuclei there. So this is the parakeratinized epithelium. Now, you have to remember, nevertheless, it's still keratinized. And where do we have keratinized epithelium? We have it in the skin, as we have discussed, thin or thick skin, right? So your vermilion border, your vermilion zone is a skin. It is a skin, it is a thin type of skin. But what is it lacking as a thin type of skin? What is not present there?
The hair follicle.
Hair follicles. You have to remember that your red zone, your vermilion zone, is a skin. It is a thin skin. Why? Because of the keratinized epithelium. But it is a very special type of thin skin. It lacks the hair follicles. There aren't any hair follicles. This is an actual question from Professor Rosa last year. Okay, how would you call this layer? And how would you call this layer? And I love this question because it shows if you understand things or not. So lots of students, they just try to memorize things and just go to the exam. They can see that. The professor definitely can see it. So you have to be able to apply your knowledge. So how would you call this layer? And just try to think about it. We have said that anybody? No?
The mucosa and submucosa.
Mucosa is with the lamina propria, with the non-keratinized or keratinized epithelium, you are right. But we have said this is a thin skin, right? This is a thin skin. So how would you call this layer? You know it. Skin, thin
Dermis.
skin, epidermis, very good.
Epidermis.
This layer right here, this is the epidermis, and this layer is the dermis. Okay, so it is true. Mucosa, submucosa, very good, very good. Just the more proper way to describe it, because it is thin skin, is that this layer is epidermis and the lower is dermis, okay? Do you see that there are protrusions into the epidermis, right? Those protrusions are called papilla. So again, do not confuse those papilla with the tongue papilla, okay? So do not confuse them. Here it is something else. So those papilla are dermal protrusions, right? So dermal protrusions into the epidermis. What is the importance? They contain lots and lots of blood vessels, okay? So those areas right here are going to contain lots and lots of blood vessels, okay? So think about it. Why do we call it red zone?
Because of the blood vessel.
Because it's more or less on the pinkish side, but again, it so depends on your tone. Actually, there is no red color. Not a single person worldwide has a natural red color to the lips. Still, it has a little bit of a pinkish undertone to it. Why? Because you will see the blood vessels. The blood vessels are very, very superficial. Unless you're living somewhere close to equator or somewhere where sun shines most of the days, so your melanocytes are actively making melanin, so that way your lip color will also change, okay? So it will change, so it will not be pinkish, it will be more on the darker side because you need the protection from all of the UV that comes from sun, okay? So again, if they ask you this weird question, why do we call it red zone? We call it red zone because the blood vessels are located within the papilla, and as you can see, they're very superficial. I'm not sure if you have hit that point yet in this uni. I have, in my 6 years, I have several times hit this point where you find yourself in this uni actively biting your lips out of stress, okay? You're so stressed that you bite it to the blood almost, okay? They're so chopped and I don't know, so like it's complete disaster. So if it has ever happened to you, you would have noticed that little tiny bite and you already bleed like no tomorrow. Why? Because the blood vessels are within the papillas and they're very, very, very superficial. Okay, so extremely, extremely superficial. So again, color depends on your melanocytes, that's all. So if, again, you're living somewhere up north, not as much sun available, the color will be paler. If you're living closer to the area where the sun shines most of the time, melanocytes will be active. They will make melanin to protect you from that sun, so the tone will be darker, that's all. Okay, finally, last thing for today, we're gonna start with the tongue, okay? So tongue, anatomical view of it, what can we see here? So first of all, the superior surface of the tongue, this one, we call it dorsal. Okay, so the visible surface of the tongue, we call it dorsal. Everybody knows who Shortzol is? Yes. Famous question. Why? Why is the superior surface of the tongue dorsal? Anybody dare to ask, to answer? To try to answer? Why do we call it dorsal? Why is the superior surface of the tongue dorsal? In order to answer this question, you need to know what does it mean, dorsal.
At the back of your mouth.
So yes, but what do you mean the back? Like This is the tip. This is also dorsal. So what does it mean, ventral dorsal? Come on, guys, you learned it last semester. Try to answer ventral dorsal. What does it mean? For sure, you have, as somebody have said, front and back, anterior, posterior, right? So that's what we usually use it for, ventral dorsal. But there is a hidden meaning to the word ventral. For example, why is this ventral surface? Why is palmar the ventral surface?
Because in anatomical position, it's facing forward.
The facing what? Facing the body, facing the core. Ventral means closest to the body. That's what it means. Ventral means closest to the trunk. So if I stick my tongue out like this my tongue is out, okay, like this. So sorry, not a good anatomical representation, but you get it. This one, is it closest to the trunk or no? The trunk is here. I stick my tongue out like this. Is it closer to the trunk or no?
No.
Yeah.
Guys, I stick my tongue out. Which one is closer to the body? Up or down?
Down.
The under the tongue. Okay, so under the tongue is closer to my body. So which one is the dorsal? Superior surface or inferior surface of the tongue?
Inferior.
Inferior is closer to the trunk. So inferior is ventral. So under your tongue, this is ventral. But your tongue, the superior surface, this is dorsal. So do you understand the answer to this question? So if he asks you the stupid question, why is the superior surface of the tongue dorsal? How would you answer that question?
Further away from the tongue.
Because dorsal means that it is facing away from the trunk. Okay, it's facing away from the trunk. That's it. So the dorsal surface of the tongue can be divided into two, into the anterior 2/3 and into the posterior third. Okay, so anterior 2/3 and posterior third. What is the division? A V-shaped structure, which is going to be known as the sulcus terminalis. Okay, so sulcus terminalis is a V-shaped depression that kind of divides your dorsal surface of the tongue into the anterior 2/3 and posterior third. At the apex of the sulcus terminalis, there is a foramen, which is called foramen cecum. Do not forget, in your skull you had foramen cecum. Here you also have foramen cecum. In embryology, we will talk about it. From the embryological perspective, this is a very important structure on the tongue. It tells you, if you have it, that thyroid gland has descended, because originally that's where the thyroid gland appears in embryo. It's right there at the root of your tongue. If you have foramen cecum, that means that thyroid gland is where it should be. If you don't have it, that means that thyroid gland is not where it should be, okay? But again, in embryo, we will learn more. So again, innervation. Innervation is the most important part for us. Anterior 2/3 of the tongue is innervated by this nerve. Which nerve is this? This is trigeminal. Okay, so this is going to be the trigeminal. So this is a cranial nerve number 5. So this is the trigeminal. The posterior third is going to be innervated by this nerve. Which nerve is this?
Ninth nerve.
Glossopharyngeal.
Glossopharyngeal. Okay, so the posterior third will be innervated by the glossopharyngeal. The most posterior, like where the epiglottis and all of this, will be innervated by this. Which one is this? That's the vagus. Okay, so cranial nerve number 10. This is going to be the vagus. Now, when we talk about the tongue, as you remember, it's covered by the specialized mucosa. Do not forget that the specialized mucosa has lots and lots of protrusions. How do we call those protrusions? We call them papillas. Those papillas are very special because they possess taste buds. You need to remember that there are 4 types of papillas. They are called filiform, fungiform, circumvallate, and foliate papillas. Okay, so 4 types: filiform, fungiform, circumvallate, and foliate papillas. Very important to remember, both anatomy and both medicine and dentistry. Okay, so both you guys should know: papillas, filiform, fungiform, circumvallate, and foliate. Now, out of the 4, filiform are the most numerous ones. Their shape is like a claw. Okay, so they're like this. Okay, so they're conical or like a claw shape. They're the only papilla which do not have taste buds. Okay, we have said that papillas are protrusions of the specialized mucosa. They're so special because they have the taste buds, but one of them, as an exception, the filiform, does not have any taste buds present in it. So what is its job? It has a mechanical role. Have you ever seen the tongue of a cat? Or like a dog? Have you ever seen their tongue? It's covered with those claws. Not claws, like conical shape kind of structures, right? The tongue has lots and lots of protrusions like this. What are those protrusions? Filiform. Why cats have such evident filiform? Because 23 hours in a day, they groom themselves. So their filiform papillas are extremely, extremely evident. We, thanks to evolution, have a shower. So we do not need to groom ourselves like this. So we just go and take a shower, hopefully. So this is why our filiform papillas are not as evident. It doesn't mean that they're not there. They're just not as prominent as in cats or dogs or any animal that grooms itself. Okay, so this is what you have to remember there. So filiform, they have a keratinized epithelium because they have a mechanical role. Which particular one, God knows, like, I don't know, clean your oral cavity or something like this, but it's not a question, so don't worry about that. Just remember, because it has a mechanical role, no taste buds. Fungiform. Fungiform are white spots on your tongue. So homework, after we're done, go to the mirror and try to find those white spots. Those white spots are the fungiform papilla. Why do we call it fungiform? Because they look like a mushroom. Okay, so they look like a mushroom. Majority of them are at the tip of the tongue. Okay, they have the taste bud. Another ones are foliate papillas. They are located on the lateral side of the tongue, and they are parallel ridges. Okay, so they're parallel ridges. Fun fact, majority of the older people seem to have more prominent ones compared to the younger people. So if we take a look at ours, and I don't know, 75-year-old guy papillas, foliate papillas, you probably won't see yours, while they have very, very prominent ones. Okay, why? No, nobody knows. And last ones are circumvallate papillas. They are huge. Do you see? So huge that if you're bored, you can actually count them. How many do you have? And if you were to count them, you'll count approximately 8 to 12 of those papillas. They are located anteriorly to the sulcus terminalis. Okay, so anteriorly to the sulcus terminalis. They're super, super big, and they are the ones which are actually responsible for a new taste called umami. Okay, so this is a 5th taste, umami. It's actually quite interesting. We have Japanese, right? In this group, I think, but I might be wrong. Okay, doesn't matter. So according to Japanese, the ones which are born and raised in Japan, umami is meaty, like a meat taste. According to American Japanese, it's delicious. So the 2 different Japan languages that you have American Japanese and you have like native, like Japanese, and the meaning is different. Okay, one of them says delicious, one of them says meaty, but just fun fact. I'll talk more about them in a sec. So take a look. Claw shape. Which kind of papilla is that?
Filiform.
This is going to be the filiform papilla. Do they have taste buds?
No.
No. Keratinized or non-keratinized epithelium? Filiform. Mechanical shape, mechanical function. Of course, it has to have keratinized epithelium to protect you from whatever damage you are doing. Take a look. The shape is more or less like a mushroom. So this is the fungiform papilla. Has the taste buds?
Yes.
Yes.
Yes. Where is it most numerous?
On the tip.
At the tip of the tongue, I heard it. And now circumvallate papilla. Okay, so now it's going to be circumvallate papilla. So how do you know that you're looking at the circumvallate papilla? They're surrounded by those deep grooves. Okay, so you can see that they have super deep grooves right here. Another name, moat, sulcus, invagination, whatever. What's the problem with this groove? Why is it not good to have those kind of grooves here? Why you wouldn't want to have them? Think, on the tongue, there are deep grooves here. Why is it a disadvantage?
The accumulation of bacteria.
Things definitely will accumulate here, like dead cells. And if dead cells accumulate here, who would like to join the party? Bacteria. If bacteria accumulates there, that will cause inflammation. Do you want that to happen? Of course not. So we need to clear those grooves. This is why under the circumvallate papilla, you will always find serous salivary glands. You will always find them there. And those special serous salivary glands are going to be known as the von Ebner's glands. Okay, so von Ebner's glands, they are very special serous salivary glands. They are located under circumvallate papilla only. And what is their job? To wash the moats, to wash the grooves, like a hose, okay? Like just clear everything out. Serous salivary glands. Does it make mucus or watery saliva?
Watery saliva.
Watery.
Very good, because their secretion is protein rich. So this is why you would want to have a watery saliva and wash those clefts right here, okay? Now, von Ebner's glands are salivary glands. So of course, they're exocrine glands. So of course, you have to have a duct that is going to be basically clearing this entire area, okay? Now, foliate papillas, just parallel ridges, etc. Now, taste buds. Typical question is to draw a taste bud. Draw me a taste bud. What is the shape? Onion shape. So you will draw it like this, like ellipsoid. Okay, so onion shape, like this. The most apical part of a taste bud is called pore. So this area right here, this is going to be known as the pore. Okay, this is where the macromolecules of the food comes in. Which cells are making up the taste bud? We have 3 types of cells: receptor cells, supporting cells, and basal cells. Okay, receptor cell is the most important cell. Another name for it, it is also known as the hair cell. Why do we call it hair cell? Because on its apical domain, it has protrusions. Those protrusions are called microvilli. From the first semester, what is the job of microvilli?
Absorption.
Full sentence, please.
Increase the surface area.
For absorption, bravo. Okay, so this is the sentence that the anatomy department wants you to say, that the microvilli, they are going to be increasing the surface area for absorption. So think about it this way: the more I chew, the more the food kind of becomes smaller into the macromolecules, right? So the more microvilli I have, the more they can absorb that macromolecules. Now, receptor cells are neuroepithelial cells. They always ask you this, always, in SCT and in the oral. Okay, which kind of cells are receptor cells? They are neuroepithelial cells. What does it mean? Neuroepithelial cells are cells which are epithelial cells, but they have a properties of a neuron. What can they do? they can synapse. From biology, everybody understands what does it mean, synapse? What does it mean, synapse?
To send the information.
Exactly, that's when the two neurons connect and they just send the information along the pathway. So here we have the nerve fibers, right? Those nerve fibers will synapse with the receptor cell. How are they capable of doing it? Because receptor cell is a neuroepithelial cell, so it can easily synapse, right? Which nerve? Facial nerve, okay? So this is very important. Also glossopharyngeal and vagus, okay? So those are the nerves which can synapse. Now we have the supporting cells. Guess what do? They just support the receptor cells, and we also have the basal cells. Basal cell is a stem cell which is going to be responsible for kind of giving rise to both, to the receptor cells and to the supporting cells. So do you have any questions here? I hope not. Typical question in the anatomy room, they will ask you. In histo, not so much, maybe only in the oral, but in the anatomy room, they ask this, that taste buds are limited to the oral cavity. They're limited to the tongue. Is this true? It's not true, okay? So taste buds can be present some other places. For example, the soft palate, the glossopalatine arch. So we have drawn it today, okay, with your uvula like this, sorry, okay? So with the uvula hanging down like this and other arch right here. Posterior surface of the epiglottis, okay? So last year, Professor Rosa asked this weird question, what is epiglottis? What is it? Everybody knows what epiglottis is?
Yeah.
So what is it?
Cartilage?
It is elastic cartilage. She wasn't happy. Any else? Any other answer?
It prevents you from choking on your food.
Yes. Wasn't happy. She was like, yeah, that's what it does, but what is it? No? Anyway, my student eventually said it because he remembered me saying it. It's a flap. That's what she wanted. It's a flap. Everybody knows what flap is? It's this, okay? So it's like opens, closes, opens, closes, open, closes. So that word was stuck in her head, and until you said it, she wasn't gonna be happy, okay? So it is a flap. So epiglottis is a flap that prevents you from choking, that covers trachea, whatever. Posterior wall of the pharynx, we'll talk about it as well. Okay, so just some taste, nothing interesting. So finally, we go for the slide. So this is gonna be our tongue. Until here, this is a dorsal surface. So everything that you can see here, this is a dorsal surface. Everything below it, this is a ventral surface, right? So again, dorsal is superior, ventral is inferior. What is in the core of the tongue? What is in the core? What is inside?
The skeletal muscle.
Very good. So this is a skeletal muscle. And you need to remember that skeletal muscle here runs in 3 directions: horizontal, vertical, and diagonal or oblique. It's the same thing. Okay, so horizontal, vertical, and oblique or diagonal. What are those?
The salivary glands.
Are they mixed, mucous, serous? What are they?
They're mixed.
They're mixed because you can see the paler acini and the darker acini, right? So the paler acini are mucous, the darker are serous, and the duct as well. Now, if I ask you this question, what is this? How would you answer this? All of this.
Epidermis.
No, epidermis is the
Epidermis?
skin.
Oh.
Sorry?
The mucosa.
Yes, but which kind of mucosa is this?
Specialized mucosa.
Very good. So this is a dorsal surface, this is a specialized mucosa. How do we call those protrusions?
Papillae.
Papilla.
Papilla, very good. So those ones, those are the fungiform papilla. And those ones here, those are the filiform papilla. Okay, from down, the ventral surface, how would you call this? Does it look the same? Does it look the same?
No.
Yes, no?
No.
No, it doesn't look the same. I can tell you this. This is way thicker. There's some other structures. It does not look the same. So can it be specialized mucosa?
No.
No, because they do not look the same. This is specialized mucosa. So what is here then?
Just lining mucosa.
This is a lining mucosa. So to make it a bit easier, the superior surface of the tongue, right, dorsal, this one, this is going to be the specialized mucosa, the most superficial part. But under the tongue, do you have any taste buds? No, so it's just lining mucosa. Okay, so this area right here, this is a lining mucosa. In the SCT, they asked this last year. Okay, so they pointed here and they asked you which kind of mucosa is this, and there was an option of saying specialized. Okay, but it isn't. This is not specialized. There aren't that many taste buds there. There's none of those things. So this is a lining mucosa. This part almost never comes up. I still teach it, but this area, they never asked this. They're more interested in this area, this comparison, okay? But nevertheless, those are the fungiform papilla. They do have a taste buds, and those are the filiform papilla. They do not have a taste bud. You can see the keratinized, highly keratinized tips, that's it. We have talked about salivary glands. I don't wanna talk more details about it. Just mixed, can you tell me what is that? Wait, I saw it here. I want to ask you. Yes, there you go. This one. What is this? How would you call it?
Demilune.
Yes, this is the serous demilune of the Italian guy who came up with this. How did we call it? Starts with G.
Giannuzzi?
No? Very good, of Giannuzzi. Okay, so this is going to be the Giannuzzi's or Giannuzzi's demilune. So this right here, this is a Giannuzzi's demilune. This is a mucus acinus with a serous cup. What is it? Is it a functional acinus? What is it? Giannuzzi's demilune, what are they?
It's artifact.
Very good. It is an artifact. It is an artifact. What does it mean, artifact?
A mistake during.
A mistake, exactly. It is a histological mistake. Okay, so a histologist was making the slide. He did not prepare it with care. So one acinus pushed into another, and as a result, Giannuzzi demilune appears. Get familiar with this structure as well. This is a nerve. Okay, it's very unfair to ask you, but they probably will. Okay, so this is a nerve. We'll talk more about it next semester in neurohistology. Okay, so this is going to be a nerve. Final slide for today, just a little bit left, guys. This is a slide that always comes up. So this is a circumvallate papilla. How do you know that this is a circumvallate papilla? Because I can see the clefts. I can see super, super deep areas right here. This is definitely circumvallate papilla. What is this? This is a specialized mucosa. So you can see that your entire circumvallate papilla is a protrusion of a specialized mucosa. How do you call those serous glands under? How did we call them?
Von
Ebner's. Okay, so those are going to be the von Ebner's serous glands. What is their job?
To clear out the clefts.
Very good. So they have to clear the clefts right here to make sure that debris does not accumulate here. Okay. And those paler areas, which you can see, those are the ducts of the von Ebner's glands. Okay, so those are going to be the ducts of the von Ebner's glands. So those ducts will empty into the cleft right here. Okay, here you can see some skeletal muscles running in different directions, and here you can see some mucous salivary gland. Okay, so mucous salivary gland, nothing interesting. So mucous salivary gland, those are the serous, von Ebner's, some skeletal muscle, nothing interesting. This part, so this is a circumvallate papilla. This is the epithelium of the circumvallate papilla. It has to be non-keratinized epithelium. Even with this magnification, you can see the dots. What are those dots? Those dots are nuclei. So it tells you that this is a non-keratinized epithelium. What is under the epithelium? What is here? What's under the epithelium, guys?
Lamina propria.
Lamina propria. So a question for you. What is in the core of the circumvallate papilla? What is inside of it? What is in the core of it?
Lamina propria.
Lamina propria. Okay, so lamina propria is in the core of the circumvallate papilla. Where are the taste buds? Within the epithelium. Do you see those paler areas, right? Those paler areas are the onion-shaped taste buds. Here we have the cleft. What are those structures?
The von Ebner gland.
Von Ebner's serous glands. What are those?
The duct.
The ducts of the von Ebner's glands, which open up here and like a hose, cleaning this entire debris here. Okay, so what are those?
The taste buds.
The taste buds. Epithelium. Which kind of epithelium?
Non-keratinized.
How do you know?
Because the last layer has
Plant cellulari has the nuclei. What is this?
lamina propria.
Lamina propria, very good. Which cells are those?
Lymphocytes.
We're in the taste bud. Which cells are those? Which cells are in the center of our taste bud?
The receptor cells.
Receptor cells. Receptor cells, what is another name for them?
The neural
Hair.
They are neuroepithelial cells, but what is another name for them?
Hair cells.
Hair cells, because they possess.
Microvilli.
Very good. How do you call this area?
Pore.
Very good. This is the pore. That's where the micromolecules of the food come in. Do you see that there are lateral cells here? What could they be? They just do this. Those are the supporting cells, okay? And this one at the base?
basal cells.
Basal cell, okay? So which cell is this?
The
Hello.
receptor cells.
So we're upside down. So which cell is this? It is at the base of the taste bud, right? So this is a taste bud like this. There is a cell at the base of it. How do we call it?
Basal.
Basal cell. Okay, which cell is this?
Supporting cells.
Supporting cell.
It is right in the center of a taste bud.
The pore. The hair cell.
The pore is just the area that is here, but there's a cell right in the center of a taste bud. What is this? This is the receptor cell. Okay, so again, if we draw a taste bud, onion shape, this area is the pore, at the bottom, basal cell, at the lateral sides, supporting cell, and wherever is in the center is the receptor cell, like this. So I repeat, what are the cells in the center?
The receptor cell.
The receptor cell. What are these cells at the sides?
The supporting cells.
Supporting.
Supporting cell.
Substances.
Supporting cell. Which cell is this?
Basal cells.
Very good. This is the basal cell. Okay. Again, to hit the point home, how do you know that this is a circumvallate papilla? This area right here is the circumvallate papilla. How do you know that?
The cleft.
Very good. So the first thing that helps you to identify anything is the cleft. Once you see that there is a deepening like this, once you see that there's some kind of paler areas, what are those paler areas?
The taste buds.
Those are the taste buds. You know that this is a circumvallate papilla. So this is the epithelium of circumvallate papilla. What is this?
The lamina propria.
Propria. What are those?
The ducts of the
Those are going to be the ducts of the glands which are under. How do we call them?
von Ebner.
Von Ebner's serous glands. Is it correct to say that von Ebner's serous glands, they are present everywhere?
No.
Like they're under all of the papilla?
No.
No, they're only under circumvallate papilla. Okay, so they're only under circumvallate papilla. This is for you. Once you're done studying, make sure that you kind of test yourself on all of the papilla, what covers them, whatever, blah, blah, blah. Remember that filiform, no taste buds, and they're the only ones which have keratinized epithelium. All others have non-keratinized epithelium, okay? So this is the place where we are going to finish for today.